All right, everyone. Today's guest is Laura Hsieh. Laura, thank you so much for being here with us. Really appreciate it.
Of course.
Laura is the founder and CEO of TippingPoint Biosciences, the first company to drug the entire chromatin network. We will talk about what that means. But I also want to say that TippingPoint is the first company that has been on the podcast that Sam actually led the investment in. So Sam is actually going to be leading this podcast, and a first for her. So Sam, take it away.
Thank you. I'm really nervous to be taking over the podcast host throne, so I hope I do a good job this time. But welcome, Laura. Today we're going to be talking about all things epigenetics, chromatin, maybe a little bit about intrinsically disordered regions of proteins. I don't know if that's too in the weeds, maybe. But I also want to chat with you a little bit about what it's like to go from academia to being a founder and also spinning out a company from a very high-profile university. And so welcome. We'd love to have you just introduce yourself, talk a little bit about your background, and we'll get into it.
Yeah, so as Sam mentioned, I'm from academia, so I'm a scientist by training. I did my PhD at University of Washington in Seattle, and then my postdoc at UCSF. And a big part of my background and expertise is in understanding mechanisms of chromatin or epigenetics biology. And so while I was at UCSF, a big goal of mine and Gita Narlikar, the lab that I was in, we really wanted to figure out assays and ways to drug chromatin better than what's out there. And so we invented a platform technology to enable us to do this. And then we spun out the company into TippingPoint Biosciences, licensed the technology from UCSF, and started in January 2023. And since then, we've raised pre-seed funding and then have really de-risked a lot of the platform technology, and then are now really focused on developing assets from our platform.
That's exciting. So I love that, you're coming from this background of being really in the weeds with a very specific area of science that's been really historically challenging for people in terms of new drug discovery in this area. And we'll talk a little bit about what that means. But before we do that, just a quick two-liner about Tipping Point. You're discovering epigenetic drugs by drugging whole chromatin networks, which we'll talk a little bit about. Why has this been historically something that's challenging for companies and researchers to do in the past?
Hey, everyone. Pardon the interruption. I just want to let you know that this podcast is an effort of Hsieh, a venture capital fund Sam and I run that invests in what we call Human 3, the next epic of health, well-being, and performance. We invest in frontier areas like genetic medicine, brain-computer interfaces, longevity, and even psychedelics. So if you're a founder building on the periphery of biotech or an investor who wants to get involved, please reach out to us at hello@xeavp.com. That's hello@xeavp.com. All right, back to the show.
Yeah, so I think, for a long time, the way that people kind of viewed chromatin in general was they were kind of like these Lego block structures where DNA wrapped around. And so a lot of people didn't pay attention to the substrate. A lot of people just kind of paid attention to the epigenetic factors that bound to chromatin and tried to drug them. And so I think it was ignored actually for a while. And then as more and more research, especially from academic labs, was ongoing, it was shown that the dynamics of the chromatin structure was actually really important in configuring how these epigenetic factors scaffolded onto chromatin and affected their structure and their behavior. And so mimicking, a complex network of interactions is obviously much harder than a single protein. And I think we, a lot of times like to simplify things and then, drug things simply. But the fact of the matter is that you really need the entire context there in order to really understand how these epigenetic factors are working to drug them specifically. And so creating that context in a solution is actually pretty challenging.
Yeah, and I think that's very much specifically what it is that your platform is solving. And we'll get into that in a little bit. But before we do that, I think just wanting to kind of go off on what you said about how solving complex problems and complex diseases, you really need that extra biological context there. And for those listening who aren't familiar with how we develop drugs today, we essentially very often find a protein that becomes our drug target, and we design molecules to go after that very specific protein in a very specific manner, which works great in some instances. So what are some examples of diseases that you would say necessitate the complex approach of drugging the entire network that can't be solved with this one drug, one protein approach?
Wait, can I ask a question first?
Yes.
Sorry. What is epigenetics?
Yes.
At a high, just from the most basic building block of what you're doing.
Yeah, so epigenetics really is the how DNA folds into a 3D structure, and how those structures perpetuate specific behaviors of DNA-dependent processes. And
so it's the shape of the DNA itself, not the actual coding, but how the
Exactly.
DNA is like structured.
Yeah, and there is kind of a code based on post-translational modifications as opposed to like DNA sequence. And so those post-translational modifications often dictate what type of, chromatin structure you're a part of. And some of those actually get what's called epigenetically inherited. So it's not just something that lasts for one cycle, but it can last for, a few generations.
Mm-hmm.
Interesting. And so the relationship between epigenetics and chromatin, is it the same thing, or is it like chromatin is a specific like thing that happens in epigenetics? Or
Yeah, so chromatin is the structure, and I think a lot of times when we talk about epigenetics, it's kind of like the part where we talk about the histone code and the inheritance, right? So if you create a typical a particular type of chromatin state, a lot of times you will maintain that state because there's a lot of feedback mechanisms, and so that epigenetics will carry on for multiple cell cycles.
So I guess now I'm trying to kind of take a step back and make an analogy. If we have our DNA sequence, we have our code, so the different types of molecules that make up DNA are kind of arranged in a sequence, and DNA can interact with itself. And so it folds into itself, but also around these things called histones, I believe, right? And so can you explain how you go from the DNA wrapping around the histones to chromatin, and then I guess epigenetics is the domain with which all of these molecules are operating in? Is that kind of what you were asking? And maybe Laura could explain that.
yeah, I guess I was just trying to understand, like for someone who maybe took biology in high school and like half paid attention, what is the best way for them to like think about this? Because there's
So I think the best way to kind of think about it is, the DNA sequence is all the same in every single cell of your body. So what differentiates a skin cell from a brain cell, right? And it's really how that DNA is actually packaged. And so the packaging dictates, which genes are actually turned on and turned off. And so if you can imagine, the DNA wraps around these histone proteins, and then if it's really compacted, then they're inaccessible to transcription factors that can turn genes on. And then if they're open, then they're more likely to be bound to transcription factors that turn genes on. Now that's just one example of a DNA process, but there's a lot of other DNA processes that are dictated by that packaging, and then perpetuated throughout generations, so that it can maintain a specific state, and that you can continue having like skin cells becoming skin cells.
And just to try to put like an analogy together, I'm sure this loses a lot of the detail, but if you had a t-shirt that had something on the front of it and it was folded in a messed up way, you couldn't really see what was on the front.
Exactly.
And if someone tried to make a copy of that t-shirt while looking at that like bunched up t-shirt that was at the bottom of the hamper, they wouldn't be able to actually replicate it in the right way because they couldn't really like see it fully. Maybe. Is that close-ish?
Yeah.
Directionally correct? Okay.
Yeah, that's kind of, yeah.
Maybe, sort of. Okay.
Yeah, I think that's good for sure. That's a good analogy. And
that can cause diseases, obviously. When
Yeah.
we're talking about the chromatin in the DNA.
Yeah, I think understanding this is probably, it kind of ties into my question, because actually stepping back for a second, the name Tipping Point, when we first met, you were telling me it was inspired by taking a cell that's in a disease state and tipping the scales of its environment to make it healthy again. And usually, this isn't like a very simple reason. There's a complex network of things happening. And so taking that into context, and basically what Brom kind of just asked, like, what is the basic mechanism of like chromatin structures and dysfunctional chromatin? How would you say that diseases that have dysfunctional chromatin differ from diseases that don't? And why is it that some diseases are more complex in nature than others in this sense?
Yeah, so I think, there's very clear examples where there are like genetic mutations, so mutations in the DNA sequence that contribute to disease, right? And a lot of studies have shown if you can correct that mutation, then you can stop a lot of those disease phenotypes. So there are examples of those diseases, but there's also a lot of examples like in cancer, in a lot of CNS disorders, where it's not just a simple mutation, right? There's a lot of different factors ongoing, and a big, I guess, like proportion of what changes cell identity is really dictated by this chromatin and epigenetics. And so it's involved in, ways that that are outside of just gene sequence. So as an example, a lot of times in cancer resistance, you sometimes don't actually see much DNA mutation anymore. It's the epigenetic or chromatin structure that's changing, and that's what's sustaining those resistance mechanisms.
And so it's like the way it's being packaged is changing, and
Exactly.
So some parts are unwinding and being open to transcription factors, and some parts are not, and that's influenced by dynamic processes that are downstream of the mutation.
Correct. Yeah.
Okay.
Yeah.
That makes a lot of sense. I mean, I think for some people who are generally familiar with epigenetics, the one thing that they may think of is, I think there was like a twin experiment in grade 11 or 11th grade biology that people kind of reference where the twins have the same DNA, but it was shown that they had a different epigenetic inheritance, but also their environment may have changed it. Do you recall that example? Maybe you could go over it for us a little bit.
Yeah. So exactly as you're saying, as because there are environmental changes or environmental differences that people are exposed to that can certainly affect the epigenetics, right? Because a lot of the post-translational modifications on histones as well as epigenetic factors, they're related to metabolism, right? So in order for a lot of these processes to occur, you need energy, right? And so a lot of these things change depending on the environment. So certainly if you have the same DNA sequence, but if you have different environmental factors, then you can have different types of chromatin structure.
Interesting. Do we know what the part of the word epigenetics, like that part that means? What does epi mean?
The epi?
Yeah, what is epi? Because we know genetics is referring to your genes, which, is dictated directly by your DNA. But do we know what the etiology of epi is?
The etymology?
Etymology, etiology. I'm like disease etiology.
Doesn't it mean higher level, like epitome? Is that where
Epitome.
it's the same root word?
Interesting. I actually don't know.
Yeah, I don't know what the, I guess the formal definition is, but I like to think of it as the layer on top of the DNA sequence.
Ah, interesting. Epi.
We can ask ChatGPT live.
ask.
What does epi mean in context of epigenetics? What is that prefix epi? It doesn't even respond. It doesn't even know. Can you believe it? How embarrassing.
We could just
What
look
does
really
epi
quick.
mean in the context of epigenetics? What does the prefix epi mean?
Epi etymology, not etiology.
That we try to be tech forward and then nothing.
So from Greek, it means upon, near to, or in addition. So I guess that makes sense. But kind of, okay, so we got into the weeds a little sooner than I expected because that's my tendency. Key difference that I need to kind of maybe work on a little bit. But when we're talking about what Tipping Point is leveraging, the tech that you licensed, that you worked on before you licensed, can you explain a little bit about how that's enabling more efficient drug discovery and what exactly it is.
Yeah, so oftentimes in targeted drug discovery, when you isolate single proteins to develop assays, you're really targeting some of the more defined structural regions, right? So some of the conserved regions. And what that means for the epigenome is that oftentimes you're targeting these epigenetic factors, and they have a lot of roles both in disease and normal cells. That's why it's really difficult to drug specifically in disease, because if you drug these conserved domains, they have functions in everything. And so really the question is, how can we find better ways to drug the epigenome more specifically? And so the way that we kind of think about it is that the context by which these epigenetic factors actually behave and scaffold onto chromatin is very different depending on specific chromatin cues and binding partners, right? And so the structure changes. So there's a lot of interfaces that become different in the context of disease.
And so these are these protein interfaces? Are they?
Yeah, so they're protein interfaces or protein DNA interfaces. And a lot of those things change in the context of disease. So you can get actually unique interfaces if you can recapitulate the context. And so that gives us the actual opportunity to now drug these epigenetic factors specifically for disease as opposed to, in everything.
Yeah. I remember we were talking a little bit about the work that you were doing, and it seems that the lab that you were working in, Geeta Narlakar's lab, you two were basically working on purification methods for some of these interfaces that really enabled structural characterization through partnerships. And just to kind of clarify again for those listening that aren't as familiar, usually when drug companies are going after a very specific target, there's already a very well characterized picture of the protein or the molecular region that's being drugged. And one of the things that got us really excited was the fact that you're working on the frontier of what these molecular structures are. And so is it safe to say that you're essentially characterizing regions that have never been drugged before?
Yeah. So that's kind of the way that we think about it, because like you said, a lot of the current structures that are out there, they're structures that have the pictures of the regions that can be drugged. And anything that's unstructured, it's really hard to predict that structure. And so it's really difficult to drug those regions. Now, if we put things into context, those unstructured regions then have different types of interactions and have the ability to become structured, right?
When you say put it into context, what does that mean, I guess, for the non-biologist?
Yeah, so a protein that has a flexible region, right? When it comes into contact with specific interactions, that flexible region can become more rigid. Yeah. Right. And so if it becomes more rigid and we understand that structure, now they can become more druggable.
I see. Yeah, I guess. And I don't know if this is a correct analogy, and correct me if I'm wrong, but when it comes to just classical molecular docking, a lot of it is based on static protein models, which is not exactly the correct, necessarily real environmental structure that you're looking at, because you don't necessarily know what the physicochemical environment is going to look like at any given time. And so it sounds to me like what you've developed is a way to purify a region that gives you the most accurate picture possible, or maybe not, I don't know, of this very hard to image and characterize region. And so if we kind of step back a level for a second, when it comes to your platform, so you have this novel assay that you developed with Geeta that you've licensed from UCSF. How does that turn into a high-throughput screening platform? What is a high-throughput screening platform, if you want to describe that to everyone? And how does that help you find new drugs?
Yeah, so the basis of our platform really comes from this discovery from Gita's lab, where she discovered that the chromatin has this intrinsic property to phase separate into biomolecular condensate. So kind of like oil and water. And what was really important is that in a biochemical setting, so it's all cell-free, the physical features of the condensates actually change based on the different types of interactions. So we can now profile, these different chromatin interactions or chromatin networks, and characterize their physical attributes. And no one chromatin state is like another, right? So each has a unique signature, and that is what enables us to assay activity level differences, right, in one state versus another. And so in terms of scaling, the hardest part for this type of platform is being able to purify these things in, high quantity and high purity, and then actually getting reproducibility, because as soon as you introduce some level of complexity, then it becomes a lot harder to reproduce. So as an example, for any given, protein purification, the concentrations can be a little bit off, but those concentrations are actually really important because they dictate, how things come together. So there's like a very specific critical concentration for how these things behave. And so those small differences can have big effects. And so that's where, we worked really hard on finding, reproducible way. And a lot of it is being able to purify these things in really high purity so that we can reproduce the assay and then scale it up for screening.
So just for those listening, assay, not essay, assay, just if you don't know what it is, it's basically a test, an experimental test that researchers use. You can have an in vitro assay, which means it's in a test tube or in a dish. You can have an in vivo assay, which means in animals, but it's really just a way to assess whether or not your experiment is working. And so this know-how plus this assay that you've developed is unique and exclusive to Tipping Point. You're able to quickly look at the effect that disease has on different cells on the whole chromatin network. But what that means in terms of high throughput screening now, you can actually take molecules and put them into this assay to see what the effect is on the cells.
Exactly. Yeah.
What does high throughput mean, and how do you scale this over time, I guess?
Yes, I think some people have different definitions of high throughput, right? Like you can be talking about thousands of molecules, hundreds of thousands of molecules, millions, right? And so I think right now we're in the tens of thousands of molecules, and the goal is, as we get more hands in the company and get more capital to standardize this so that we can do hundreds of thousands of molecules.
So basically you're screening tens of thousands of molecules at a time in a very efficient manner against regions of protein-protein or protein-DNA interfaces that have never been characterized before. And so you're building this flywheel of potentially useful data that can then be used to leverage computational tooling down the road. So I guess what you're doing is looking at different molecules, seeing what are hits, seeing what's not. But for those listening again, how does this translate into developing a drug program for a specific disease? I mean, we've talked about a few. I think it might be useful to maybe walk through an example. What do you think?
Sounds good to me.
Okay, so maybe it might be useful to chat a little bit about DIPG, what DIPG is, and why it's a great proof of concept for your platform specifically.
Yeah, so for those of you that don't know, DIPG stands for diffuse intrinsic pontine glioma. It is a pediatric brainstem tumor that grows in the pons of the brainstem, and it's a really high unmet disease. These kids get diagnosed and die within months of diagnosis. Since 60 years from the discovery of DIPG, there have been no FDA-approved drugs in the space, and so it's a really high unmet need, and these patients really need answers. And
so you get diagnosed and they do nothing, basically? Is there surgery or anything?
So because of the location of the tumor, surgery is not feasible.
I see.
And so right now it's mostly radiation treatment and clinical trials, right? But radiation treatment really hasn't been beneficial for most of the patients. And so a lot of these patients and the patient families are really looking for alternative therapies. And so a lot of the work that's been done in this space has really been done in academia. And now there are starting to be like a couple of companies that spun out from academia that is working in this space.
Wow. And how prevalent is it?
So DIPG is a subsection of a kind of a bigger group of diffuse midline gliomas in kids and a smaller proportion of adults. The mutation manifests in around 1,000 to 2,000 patients population, and it's really the maintainer of the tumorigenesis in this disease. So people have shown that if you can remove this particular mutation that causes the disease, then you can actually inhibit the tumorigenesis.
Interesting. Yeah, I mean, it sounds and is very devastating. I think what was very shocking to me is the very simple biological dysfunction that causes such a devastating phenotype. Can you talk to us a little bit about, is it like the HEC27, like triple methylation that happens? Is this what it's called? I'm not sure if I'm remembering it correctly.
It's H3K27 trimethylation. Yeah,
Okay.
so it's a histone methylation mark. And so what happens is there's a histone mutation. It's a single amino acid mutation, and it's found in greater than 80% of the DIPG population. And for like the smaller percentage, there's oftentimes something that mimics the H3K27M mutation. And so what happens is the mutation actually incorporates in only 5 to 10% of the genome, but it interacts with one of the major epigenetic proteins. It basically inactivates it and causes a demethylation event globally. And so you get, changes in gene expression that, relate to the tumorigenic phenotype.
Jeez. That's wild. So basically that's like, I guess, a testament to how ubiquitous like the effect of some change in the whole chromatin network can be.
Exactly.
Interesting.
So the way that I kind of, think about it is that a lot of these epigenetic states, they're always in competition to become something else, right? So a lot of these states are poised, and so you need to continue to maintain these states in order to perpetuate the DNA process, right? And so if that gets disrupted, then other things can come in and invade and take over. And so our name, Tipping Point, I mean, it's a really fine tipping point between the balance of, how you establish these epigenetic states. And so since things are always kind of in an equilibrium where things are dynamic in chromatin, can we use, these modulators to bias it in a particular state versus another?
And push the equilibrium to the healthy state.
Exactly. Yeah.
Interesting. I mean, I have so many different avenues that I can go down.
We were talking about the process of developing the drug. I think that's where we started.
Yeah,
right?
and I think so we talked about what makes the platform different. So you essentially would, for example, in the case of DIPG, you would try to replicate these cells in your platform and then start running drug screens against it. And essentially what Tipping Point is able to do is capture the intrinsically disordered region that causes this mutation in the most accurate way possible. And so in theory, you hope that your drug that you find and develop would be able to fix that mutation better than any other drug out there.
Yeah, so I think the difficulty of targeting this mutation is that it's only a single amino acid change of a major, chromatin protein. And so if you purify it, like on a single protein level, there's no way to tell the difference, especially because the mutation is in an intrinsically disordered domain. There's not really a structure. And so the thing about this, the reason why we picked this particular disease is that the mutation in context with the surrounding chromatin environment is what basically inactivates the enzyme, and you need everything there to get maximal inhibition of the enzyme. If you only have, two of the proteins, it doesn't mimic the result, right? So this is a perfect example where you need a lot of that complexity in order to actually mimic what happens in a cell. And so our platform is able to, for the first time, target this mutation in the right context. And the idea is that if we can mimic the disease, we can find modulators that specifically disrupt the disease interactions, and then we counter screen with the analogous healthy state, so without the mutation, to then find then confirm that the molecules don't affect the healthy state. And so from the very get-go, we're already, we're optimizing for specificity.
Sorry if you answered this already and I didn't hear it, but do you basically have to get cells from people that have DIPG to make sure that your model is representative of the actual disease?
So no, because our platform is all biochemical based. So we reconstitute everything. And the beauty of the biochemical assay is that we get to customize it the way that we want. We, of course, characterize the patient cells, and then we mimic it biochemically. But we also get to control what goes into our test tube, so that when we do target deconvolution, then we can, it's not targeting an entire cell, and it's not like hundreds of proteins. It's basically 2 macromolecules that we can really look at.
And then, so that's the model. And then where do all these candidate drugs come from? Are they invented by an AI? Is it a screening library that you download and run through?
So right now,
combo of both?
Yeah, so right now, because computational methods are really bad at predicting a lot of this context, because we don't have any of the structural data, we are basically using small molecule libraries and doing everything in a wet lab setting, right? But the idea is that as we develop our pipeline, right, so we get these hits, we confirm that they have, activity, the wanted activity in cell lines, and then as we deconvolute the target and do the structural work, we can plug that into, some of the AI software that can help us then predict what molecules will actually work in these different types of contexts.
Okay, I see. Interesting.
Yeah, I mean, I'm really, one of the things I want to call out is that we just kind of talked about on a recent podcast what we call human 3 and how we invest and the different characteristics that we look for in companies. And one of the things that I got really excited about, we call it endogenous accelerant. So whether a company is essentially creating an endogenous accelerant within their own company, which to us means, is the company creating not just new assets, but an armamentarium of data that they can use to really propel not just their own platform and asset development further, but like the body of work and the field that they're working in further as well. And so we feel that you're at the frontier of this area of characterizing biochemically and biophysically these regions that have never been drugged before. And that's not just going to help Tipping Point, that's going to help patients, that's going to help other people that may be wanting to look at this for whatever reasons, whether that be research or commercial. And I think it's worth noting that when you were just explaining to Brom's question, or in response to Brom's question, that you're not looking at patient cells specifically, but you're zooming in even further to get drilled down into the mechanism, that if you were to actually be looking at the cells, that's like another strategy that people take too. That's what I guess people would call a phenotypic screen in a way. If you're looking at a larger, like, I guess, organism or part of the organism, like the cell, and seeing what happens. And so kind of bringing it back to DIPG, Are there companies that have already tried doing drug development for DIPG? How have they done this drug development? Has it been in the same way that you are trying to do drug development, or in a similar way? And how do you think about that?
Yeah, so I think as a rare disease, there haven't been a lot of companies that are in the DIPG space. And what has often happened in rare diseases is a lot of people repurpose drugs, right? So drugs that they know maybe affect epigenetic structure because this is DIPG is really an epigenetic disease. Or some of the other companies have compounds that they've basically done phenotypic screens, tested their molecules in a vast array of different types of cancer cell lines, and then maybe found hits for DIPG. And so I think in academia, there's a lot of great work being done at a lot of institutions that basically are doing CAR T therapies that target some of the receptors on DIPG cells. And so that work is also ongoing. But in terms of small molecules, there hasn't been a lot of exploration in terms of developing new chemical matter.
And for those listening, small molecules are generally thought of as being preferred for drug development for a number of reasons. Can you share why that is?
So I think especially in a space like DIPG, it is because you have to get across the blood-brain barrier. And so even though we know that affecting the H3, K27M mutation, if we can like knock it out, for example, with like CRISPR-based methods, that would be great. It's really hard to deliver that into the brain, right? And so one of the nice things that you can do to modulate small molecules is to design them to cross the blood-brain barrier.
Mm-hmm. I think we were talking about a company that was acquired recently. I think they were acquired for almost $1 billion, Chimerix. They were acquired by Jazz Pharmaceuticals. You mentioned that they were developing a drug for this specific mutation. Are they... Like, what kind of drug is this? How do you think about this compared to the drug that you would be developing?
Yeah, so the history of Chimerix's drug, ONC201, it seems like, it was originally developed to go after a few different types of, a lot of different indications. And they did a phenotypic screen to find that it did have an effect in DIPG. ONC201 specifically, the mechanism is not really clear cut. It has a long history. And there's, a lot of different pathways it hits. It hits metabolic pathways. People now think that it hits like apoptotic pathways. So the mechanism is not very well known. But what has been shown in clinical trials for DIPG is that there's a proportion of DMG patients where they do see extension of life for about like 1 year to 1.5 years. And in those patients, it seems like it's changing that methylation, histone methylation level, right? So that biomarker seems to be changing, and that's correlated with these patients surviving longer, but it only hits, a small proportion of the patients. And it's, like I said, it only affects their lives for a year to a year and a half. Now that's somewhat of a win, but obviously we want to have, more far-reaching effects for like these patients.
Remission.
Right. Yeah. We want to have, we want to be able to have them survive longer. We want to, be able to hit a bigger proportion of the patients. And so our molecule, what we know about it is that it's directly on target to change that methylation. And so because we're taking a very mechanistic approach to hitting the chromatin in the right way, we're hoping that it can, have a broader effect for any patients with this mutation.
That's great. And so it's great that Chimerix was able to get this drug into patients and show that it can extend life. I think just to kind of reiterate that phenotypic screening in a way, it's kind of like looking top down. Again, you're not really designing things in a very specific manner for a specific piece of biology that's going wrong. And so sometimes you get off-target effects. You mentioned that it's involved perhaps in the apoptotic pathways, which could lead to warranted or unwarranted cell death, which could be bad. But in the sense of, going bottom up, would you say a few things on what you think, like in theory, going bottom up and doing this type of mechanistic drug design, what the full frontier is. Like, is it disease modification? What does disease modification mean in this sense?
Yeah, so I think whenever you understand the mechanism of the drug, then you're always starting at a better place because you understand, what target it's hitting and how you're affecting those pathways, right? And so if you understand the exact mechanism, it often is actually easier to understand, to maybe do like combination therapy. And for something like DIPG, a lot of the field agrees that, there may be a need for combination therapy just because it's such an aggressive tumor. And if we can understand what mechanism it's hitting, then, obviously for the other treatment that we're adding on, we can understand the exact pathways, right? And I think that poses less risk for safety, and it also gives us a better understanding of how we can make the drugs better, right? And so I think that's kind of our approach is if we can take a more mechanistic approach in drugging chromatin a lot more specifically, then we can have better control and better safety profiles.
I have a high-level non sequitur question, sort of. So I understand the benefits of starting with something like DIPG because it's rare, and so you can probably get to the clinic faster and all those benefits. But what are some other diseases that have epigenetic drivers or are driven by dysfunctional chromatin? Maybe diseases that are more commonly well known that people might be able to resonate with.
Yeah, so I mean, epigenetics really, like from the start of this podcast, we've established that it's really important for cell identity. So any disease where cell identity is changed, right, which encompasses a lot. And so I think obviously a lot of cancer, because we see that cells become more a differentiated state, go to kind of like a stem cell state in cancer. So a lot of cancers are involved. And then there's a lot of CNS diseases. A lot of people are showing that if you change the function of epigenetic regulators, then you're changing kind of the differentiation state of, things like Alzheimer's and Parkinson's, right? So there's an area there. And then there's also, now studies of like things like muscular dystrophies, right? If you change chromatin structure, you change the nuclear integrity of the cells, and that has a lot of implications for interacting with the nuclear lamina. So that affects a lot of these muscular dystrophies. And
so you think that 7 years from now or something, you might be able to be going after all of these things, or have at least a program for neurodegenerative diseases, muscular dystrophies, various cancers. It's sort of like a single technology that can be applied to all these different fields.
Yes, yeah. And eventually, aging, right? Because epigenetics is really central to the aging phenotype.
Interesting. Is that just because the folds become increasingly disordered over time as like cells replicate, or?
Yeah, so the chromatin obviously over time gets more damaged, and then the chromatin also changes structure. So if you look at chromatin structure over the course of someone's lifetime, it drastically changes. Yeah. And so there's a lot of implications in can we retain certain structures, prevent some of those aging phenotypes.
Are there any, all of this seems so new in some sense. Like, are there any FDA-approved drugs that target in this chromatin in the same way that your drugs do? Or is this like total de novo? Jeez.
Yeah, so there are no drugs that target epigenetics in the way that we do. The typical epigenetic drugs that are FDA approved go after kind of these like conserved domains in very specific disease indications.
Yeah.
And I think that part of the reason why there's been so much failure in the epigenetic drug space is because they've been going after these conserved domains that have a lot of functions in normal function as well.
I see. And so I got it. Interesting. Wow.
I'm going to try to ask like a semi-non-technical question here. I'm thinking about the future in which you are able to drug these different diseases that Brom just mentioned. And so what does this look like practically if someone gets diagnosed with Alzheimer's and let's say Tipping Point has an epigenetic drug for Alzheimer's disease, a specific form that you've been able to characterize? Does it look like someone getting diagnosed and having their own cells profiled in a very personalized way before, they're prescribed Tipping Point's drug? How do you think about that from, I guess, a long-term perspective when there are other drugs like, the antibodies that are available? Does your drug just immediately become the best in class because of the disease modification? Or like you said, is it a combination therapy based on the individual's unique situation?
Yes, I think for some of the more complicated diseases like Alzheimer's, there's definitely specific types of epigenetic profiles that are prevalent in certain proportions of patients. And so I think a lot of it is characterizing, and right now, I think, especially as sequencing is becoming cheaper and cheaper, we're getting more information from patient cells, right? Understanding and patient tissues where we're understanding what are the epigenetic marks that are changing. And so because there is epigenetics is really central to that differentiation, I envision that for something like Alzheimer's, we can create a drug that can be more applicable to perhaps a bigger proportion of the patients where we can change the differentiation state. And then it allows for a lot of things like combination therapies where then, we can take more, they can become more effective because we're changing the epigenetic state.
So it's like you're priming the cell differentiation environment to be more receptive to other types of interventions because it's less bound to that path that it's on to become a destructive cell.
Exactly.
Interesting. Yeah. That's really cool. Okay, interesting. So, let's take a step back and let's talk about how you decided that this was an area of biology that you wanted to study. So you did your undergrad, you did your PhD, and then you decided to choose Geeta's lab to do your postdoc in. What about this area was personally interesting to you?
So, I always have been interested in chromatin biology. I think in undergrad, as soon as I got into a lab, I took an epigenetic course and I was just hooked. And I think a lot of it just stems from this idea that it's this complicated structure, right, that really dictates what a cell becomes. And if we can figure out that formula for how we can manipulate the structure, then there's a lot of things that we can do with that technology, right? And so I think a lot of, my interest really stems from this idea that epigenetics controls such a big part of cell identity. And if we can do more research in this area, find more tools to figure out how to control it, then we can cure a lot of different diseases.
That's really cool. So I guess that aha moment for you was like, okay, everyone talks about your DNA sequence being your personal genetic code and you're fated to your code, but actually in reality, it's your epigenome that dictates that. And you can manipulate cell fate, if you will, by managing that.
Yeah, and the important thing about epigenetics is I think at the beginning I said there's certain states that are inherited. So it continues to perpetuate, right, over multiple cell cycles. And I think the other thing is that it's reversible, right? A mutation in a DNA sequence, you can't, reverse it with a small molecule. But for epigenetics, you push it into one state and you can reverse that too.
It's interesting. I guess in terms of where you're at now, we're all in San Francisco right now. This is a UCSF spin out. You've obviously been in the Bay Area since this started. And for the past couple of years, there's also been a lot of discourse, of course, around tech bio companies. And so do you sometimes find yourself having to explain what the difference between being a platform biotech versus a tech bio company is to some people?
Yeah, sometimes. I mean, I think, we do have, certain computational methods to do some of our analysis, but I think a lot of the computation is really for the future of the company. And we want to implement those tools when we have the data. But yeah, our platform, I think, it doesn't have a big AI component at this moment. And a lot of it is wet lab. And I think sometimes, because we're a platform, people just assume that, we have some more computational aspects to the platform.
I mean, these days it's rare to not be AI driven, and I think it makes it more exciting because it means that it's kind of overlooked, right? Because everyone's too busy chasing AI hype trains. So I like the fact that AI is not like the core story behind the company.
Yeah.
I have a random question that I thought about when you mentioned the aging bit. So a lot of the companies that we've backed, not all of them, but some of them have sort of this potential to not just heal people that are super sick, but also maybe enhance people that are already healthy. I mean, the obvious example is some of the psychedelic medicine companies that we've backed. Like a lot of people will use psychedelics just to expand their minds, whatever that means, right? At least they think they're expanding their minds. Are there any things that theoretically, not even necessarily near term, that you could do by manipulating chromatin that could give someone, some healthy person, some sort of edge in something, whether it's cognitive enhancement, whether it's like athletic performance, whether it's something along those lines? Is there anything just theoretically that is possible there? Or is this pretty much targeted at, people that have these diseases? We can now fix them, but we're not going to be able to enhance anyone.
No, I mean, absolutely. We can, and as long as it's safe, the idea is that, yeah, we can definitely enhance certain activities. I mean, epigenetics is so core to development. So you can imagine that, so for example, in terms of, like insects, there's been a lot of studies to show that epigenetics enforces actually certain behaviors of insects. It's pretty cool. So like in terms of like the ant population, there's like worker behavior, and then there's queen behavior, and epigenetics actually dictates that, right? So there's this like
the difference between a worker and a queen is an epigenetic. Based on epigenetics, basically?
Yeah, so if you change the epigenetics, it changes their behavior. So,
interesting.
Yeah. So
I want my epigenome to code for queen.
And is that something that, like, theoretically, if you were to do these experiments on flies, or maybe in some alternate universe doing this on humans, would it have to be something that was done at the time that like a person was conceived, or is it something that could be done once they're already a fully grown adult?
A fully grown adult. I mean, there's a lot of diet that actually changes your epigenetics, right? And so if we
Really?
so if we can find, the key to, like if you can change, certain types of foods you eat, it changes your epigenetics, and then it results in, specific types of, enhancements. That definitely, yeah.
Wait, so I have to ask, because you're the epigenetics expert, are there any diet things that you do for this purpose?
So I remember going to like a seminar where like depending on what type of like fat you consume, so like whether it's like olive oil versus butter versus canola oil, it actually changes your epigenetic makeup. And it's interesting because I think some of it seems like olive oil is it changes your epigenetic makeup less so than some of the other fats that you eat. And so it's interesting because once you eat it, it changes your makeup, and then you sustain it over multiple generations, right? And so
and when you say generations, are you talking generations of cells or generations of humans?
Generations in cells.
In cells specifically.
Yeah.
And do you know, are those epigenetic changes correlated with like a behavioral outcome, or like what are they correlated with?
Yeah, so
in terms of like something that you could observe.
Yeah, so it's not behavior in this case, but correlated with I think like your sometimes your metabolic outcome, right? So like when there's things like droughts and or famines, right? When you look at the makeup of some of these people, you can see that their epigenetics are actually changing.
In response to going without water for a long time or less water for a while.
Right.
Interesting.
Or not eating for a while, right?
Crazy.
So are you saying that because olive oil seems to induce less epigenetic change, that's one of the reasons why perhaps it's healthier, because you're not perturbing that state that's already fixed?
Yeah.
Maybe that's why all the people in the blue zones, well, in one of the blue zones, they're olive oil consumers, right? Isn't it in like southern Italy? And isn't that one of the blue zones?
That is, I don't know. I don't even know if the blue zone thing is real. It sounds like some pop size, but who knows? I mean, as you move away from olive oil and start consuming other things, you talk with your hands less, I think.
Yeah, that's true.
An epigenetic driver.
Oh man. I just realized also why I thought the mutation was HEC 297. It's because those are the cells that you showed me in the lab when I came.
Oh yeah, they're like the embryonic kidney cells. Yeah. We use those as a control.
You were showing me the control cells. I know that HEC cells are often used for that, but this is the H3K27M.
Yeah.
Okay.
Yeah.
I don't know. I can't remember these things, but there's always all these different, I guess, words and number combinations for genes too. The other day you were saying like you don't know how people remember all these different genes, but it's interesting. I guess in terms of where you're at right now with your company and the platform, what are you looking forward to achieving in the next 12 months specifically at a very high level?
Yeah, so right now we're really working on, a couple different programs within the company. So developing very specific assets, one for DIPG and then for, a few other indications. And we're really excited about, getting to in vivo studies and really showing that the small molecules that we're getting off our platform can translate into in vivo models.
Yeah, that's really exciting. And once you get into your in vivo models, you can show like platform validation and then eventually get into IND studies and take this into humans too.
Yes, yeah.
That's really exciting. Great. And I guess, you're doing all of this. I think we talked a little bit about this at the beginning, but I'm genuinely curious. I'm sure you're asked this all the time. What is it like to go from being on an academic track to then becoming a founder? And like, are there things that you've loved and didn't expect to love? And also, what are some of the things that have been challenging?
I mean, it's a huge transition. I think that no matter how many people you talk to and get advice from, hearing about their stories is very different than actually experiencing it. And I think that, it's just that the structure of academia and industry are just so different, right? Like in industry, you work in teams, you really have to work well together, you need complementary skills. In academia, you're a lot more independent. And so you're not used to working in teams and building, different types of like strengths within the team. And then I think the other part is just as a founder wearing so many different hats, right? I think when you're a scientist, you focus on the science. And I think even when you become a professor, of course, it's a really busy job and it's a hard job. But I think you kind of, you're not as limited by timelines and funding, right? Like you have more time, you can be a lot more exploratory. Whereas in industry, a lot of it is you need to get to this milestone, you need a fundraise, a lot of things have a lot more purpose. Things are accelerated, and I think the goals are just very different. And then you have to also, as a founder, do so many different things, right?
You have to do podcasts. I mean, it's horrible. It's so distracting.
One of the things that I have said to many students who ask me how to like get a job outside of academia that are maybe doing a PhD or a master's or a postdoc is that, at least from where I stand, I feel like, very ironically, the PhD experience actually makes you ready to be a founder, because like you're constantly reinventing your idea to try and in response to your experiments. You're constantly asking for grant funding. You're used to like failure and trying to like keep going. Like, would you say that's true in your experience in some way? I know you've just kind of explained how academia and industry are different, but the skills that you've built to try and like take your project in your PhD and keep going, has that translated to keeping the company, in good spirits and keeping the projects going there?
So I would say that it really depends on the PhD and postdoc experience that you have, right? Like I think that sometimes you go into a lab and there's a set project for you, and that's very different than reinventing something on your own and reiterating, right? And I think in the particular experience that I had in my postdoc, especially in Gita's lab, a lot of it was reinventing a lot of things. And so in that sense, I do think that a lot of those skill sets do translate well to industry. But there are other, parts, like you have to change how you communicate, right? The storytelling in academia is so different than storytelling in industry. And then I think the other part is that you have to be really decisive and good at making decisions. And I think that, and you have to do it quickly. And then you also have to be able to work with a lot of different people and with from different experience levels. And I think those are things that you don't experience much at all in academia. And so I think those things have really, it's been a challenge, but I've learned a lot from it.
That's really valuable insight. I think it sounds like Gita's lab was very conducive to this idea of you becoming an entrepreneur. I guess one of my final questions is, how did you start to access the VC network from your vantage point? Because I don't think Gita had done other companies before, so I'm not sure. It's probably a question that a lot of scientists that are listening might have. Like, how did you start to break through into the investor arena and start coming up with this idea to spin out a company?
Yeah, so I think at the very beginning, I did use Gita as a resource just because she is a really well-known PI. She's a National Academy of Sciences member. And so using her connections, so some of the PIs that started companies at UCSF, that's kind of how we tapped in initially. But I think the majority of the investor relationships that I've made, it was actually through networking, right? Like I think like as soon as I transitioned out, I was at every single networking event. Like anytime, I got an invitation somewhere, I would go. And then a big part of it is actually being at NBC, I was able to meet a lot of founders. And it's the founders that have really introduced me to a lot of investors. And especially for successful founders, those connections to investors are meaningful. And then also like exposure, right? Going to conferences, presenting, pitching, and then, being very present on LinkedIn. Those are like a lot of the different things that help me establish some of the relationships. But it was definitely a challenge because coming out of academia, I didn't have the same industry networks. And like I said, Gita knew professors, but she actually didn't know a lot of investors or people in industry. And so I had to basically form a lot of these connections on my own.
So to ask a question that would happen before the how do you meet VCs part of the startup journey, if someone is in the shoes that you were in when you were doing your postdoc and they're maybe deciding between I'm going to start a company or I'm going to do another postdoc or like stay in academia, what question should they ask themselves to help make that decision?
I think for me, my advice would be like only be an entrepreneur if you really believe in what you're doing and you can't sleep unless you do it, right? Because I think especially as a first-time founder entrepreneur that's transitioning from academia, it is really tough. And so you have to, be persistent, continue to forge forward and do whatever it takes to continue the company. And I think if you don't, have those, if you're not willing to do that, then I think that it's, perhaps being an entrepreneur is not the right path at the moment.
Fair enough. But you also have to do that to some extent in academia, right? Like you have to be sleeping and eating and breathing the problem if you actually want to be successful at it, right? Sort of.
Yeah,
Kind
but
of.
I think that so my experience is as a solo full-time founder, right? Like Gita has provided, support as my academic founder, but I had to do a lot of things on my own. And in that sense, I think that in academia, you do have your colleagues and support. You have, ways that you can collaborate with experienced PIs. And so I think it's different. It's hard in a different way, but I think that as an entrepreneur, especially if you're starting the company on your own full time, then you don't really have anyone. It's a very lonely, job. And so I think that, there's a lot of different types of challenges by, being on your own.
Totally.
Yeah, I'm just like I'm listening, and it reminded me of when I first met Brom. He was sharing his experience with me and starting the fund on his own, and it definitely sounds like it just presents its own unique set of challenges when you're going out and doing something new that no one's done before by yourself and building up that support network from scratch. Interesting. And it's interesting because we actually found you, Brom found you and Tipping Point when we were doing our search for companies. And I actually want to ask you, like, what stood out to you about Tipping Point when you were looking through companies that, made you want to bring it to our attention and our deal sourcing?
It's funny. I had a similar question to ask you. But I found Tipping Point on PitchBook of all places. Only time I've ever found anything interesting on PitchBook when it comes to startups. Not even kidding. Zero other investments have been made through PitchBook. I think it was the fact that the accelerator you had gone through, IndieBio, I was able to find your page after seeing you on PitchBook, and I've been on the IndieBio mailing lists forever. I feel like it was one of the first things I signed up for when I started the fund, but very rarely do they have anything that fits into our criteria. But I always sort of respected them as an accelerator. It seemed like they do cool things, but so much of their stuff, like lab-grown vegan leather, is just like not, it's not really like in our investment criteria. So when I saw that there was you had some focus on the brain because DIPG is a brain cancer. And I was like, oh, there's a company doing real science in brain-related areas that's gone through this accelerator, but they haven't raised like a massive Series A yet, so they're not too late for us. And it seemed interesting. I didn't know anything about chromatin or IDRs. And then I showed you, Sam, and you really took it from there. Mm-hmm. And like I mentioned at the beginning of the podcast, you more or less drove the investment. I think if I hadn't been working with you, I would have not made the investment because I would have not known how to even go about understanding what was actually being done at Tipping Point. So I was going to ask you, why did you more or less decide?
It's a great question. I think I've alluded to it loosely throughout the podcast, but I always get excited when companies are building something that can immediately translate into patients, but can also generate its own sub-domain of biology or science. And it's a very rare balance to strike when you're a seed stage company where you're going after an area where you can commercialize, you can raise from venture capital, it's a venture backable company, but where you can also contribute to the basic research at the same time. I have this whole rant that I do about how I think there's a little bit of an innovation crisis in academia, and so we must back platform companies in whatever sense of the word that are also contributing to the basic research in some way. And it was immediately clear to me that because you're at the frontier of elucidating new structures in a way that no one else will be able to me, it fit all that criteria, but also it felt like this is a company that can continue to sustain itself, both in terms of innovation, but fuel for new asset development.
So not an incremental approach.
No.
Solving a real problem
Yes.
that, as Laura pointed out, does not have an existing solution. And as they develop these initial products, they will gain new insights into biology that in theory should allow them to create more products even more efficiently and faster.
Yes, and the know-how that is required to optimize the assay that you've licensed is something that no one can replicate. And over time, that knowledge moat continues to grow. And so I'm really excited about what you're doing. But yeah, I guess maybe we'll end with a fun one. if you could go back to yourself when you were in your 20s, let's say 21, and tell yourself one thing, what would that be?
My 20s, wow, that's a while ago. I think that, I think that I wish in my 20s, I would have taken a lot more risks, like starting this company, right? I think, there were a lot of risks to start this company, but I really believe in what we're doing and that we can change how drug discovery is done. And so I think that, I wish I would have been, more bold when I was younger, so that, I can have just more experiences.
That's awesome. I think that's definitely good advice for anyone, but I think that you're doing amazing things and really excited to see where this goes. Before we go, do you have any other questions for Laura?
I have one more question, and I'm sorry to extend the podcast.
No, don't say sorry.
Thinking back to when we first had a call with you, like I guess it was last June, probably almost a year ago. Mm-hmm. I feel like you've gotten significantly better at pitching and presenting Tipping Point since then.
Oh, thanks.
And I remember we had this conversation where I was like, conceptually, this seems good. She could probably do a better job of selling the vision, which is probably going to help her in the future. How did you get better? Did you make a concerted effort to, or did you just kind of naturally get better as a byproduct of talking to a lot of investors?
Oh, no, I definitely made a concerted effort. I think anytime I, approach a VC, no matter what their answer is, I try to take feedback, not personally and kind of as a learning experience. And so since I've pitched to you guys, I probably made like 100 more variations of my pitch. And I've just been, working with different founders, mentors, investors to just continue honing in on a message that, is more, marketable and also, resonates with a lot more people. And so I think, a lot of these, the advice that I get from different people has definitely helped me. And obviously, like I spend a lot of time on the pitch deck. That's like a big portion of my time. And then also practicing it.
Well, yeah. So are there any like actionable insights or things that you thought about when you were going through this process of refining the pitch that could be applied to anyone else trying to get better at pitching?
Yeah, so I think, pitching to different audiences kind of helps you capture the different perspectives that people people want to hear different things. And I think practicing different renditions of that really helps you kind of understand, okay, what's working, what's not working, and, how is the best way to present this? And I don't know if, my pitch now is is the best form now. Maybe I, make an epiphany and make a,
it's
different
never
change.
The final form.
Yeah,
it's always
but I think that, getting the different perspectives has been, really helpful.
So by that, you mean pitching to different types of VCs, maybe pitching to people that are not VCs and just seeing all the, like triangulating the truth from all of their different feedback, more or less?
Yeah. And taking their advice and actually doing it, right? Like, I think like sometimes I hear this a lot where people say, oh, like there's a lot of different, opinions. Like as a CEO, you have to pick and choose what opinions, you want to take. But I think that, for particularly pitching, trying different variations of it has been really helpful.
Fair enough. The classic A/B test.
I was going to say, I think just it's especially admirable that you've been able to improve the pitch and communicate such a complicated concept, because this isn't an easy, intuitive concept to talk about, as I'm sure everyone listening can attest to after listening up to this point. And so I guess, boiling that down do you sometimes feel like, and now I'm extending the podcast, but do you sometimes feel like you kind of have a fear of not saying enough and you just don't like, or has that kind of been something that you've optimized over time? Do you know what I mean? Where it's like, how do you gauge what the right level of technical is good for this individual? Do you generally spend a couple minutes talking to people at the beginning to decide which rendition to pitch, I should say?
Yeah, I mean, understanding the background that they come from is definitely helpful, right? For, some of the non-science folks, I'll probably, be less technical. And then for the science folks, I'll be more technical. And so I think, understanding their background definitely helps with that.
Okay. Do you have any other questions?
That's it.
All right. Well, I hope I did a decent job of hosting. Thank you for being my guinea pig with the hosting.
Thanks, Brom and Sam, for hosting for having me on this podcast.
Yeah, it was fun. Well,
Where can people find you on the internet and find Tipping Point on the internet?
So we have a website, TippingPointBiosciences.com, and then for me, probably LinkedIn is the best way.
Right on. All right. Thanks, Laura.
thank you.
Thanks.